All right, so we are group window back 3 to 6 lab, and our topics on how viruses direct evolution of life. And we will be focusing on the buzz about cucumber mosaic virus. I am Jotham Heystee.
I'm MaryClaire Allen.
I'm Izabella.
The history of organisms' interactions with viruses is long and complex. In fact, all known species live with many viruses that colonize their cells, collectively known as the virome. Of course, the relationship with these intracellular parasites has shaped the evolutionary history of life. As certain viruses evolved virulence factors and other methods of causing disease amongst their hosts, organisms have necessarily evolved immune systems to defend themselves and halt viral replication. In turn, viruses evolve responses to evade the immune system. But viruses and hosts have influenced each other's evolution in other ways as well, in some cases forming an intimate symbiotic relationship that neither could persist without.
There are quite a few ways that viruses can influence the evolution of their hosts. Viruses can alter their hosts' genomes, either by controlled insertion, as with retroviruses and some lysogenic viruses, or by somewhat random mutation, such as hepatitis B virus, papillomavirus, Epstein-Barr virus, and many other tumorigenic viruses. They can also shape the course of evolution simply by serving as a selective force for their host. Nearly every virus exerts this kind of pressure on their host, as they are obligate intracellular parasites. They often lower the fitness of at least one of their hosts for those that have multiple reservoirs. This encourages development of heritable defenses against these invaders. However, viruses can also establish symbiotic relationships.
An immune response is itself an evolutionary development to combat viruses and other pathogens. Through billions of years of interaction, complex defenses against viral invasion have arisen. From the most basic barrier defense to the most complex system of B cells and T cells in mammals, a veritable arsenal has developed to protect against viral invasion. And with each new host strategy, viruses quickly develop a response. This interplay drives the evolution of both the host and the virus.
Many viruses direct the evolution of their hosts by way of immunosuppressive qualities. For their own survival, all viruses have evolved methods of shutting down or dealing with the host's immune response. For example, in humans, the virally derived protein syncytin is crucial in forming placenta. Without syncytin, the fetal cells cannot fuse to form the placenta, and the baby will always die. As it turns out, the syncytin gene was acquired from a retrovirus infection and was once part of a retrovirus envelope protein. It is thought that the original viral protein aided the retrovirus in suppressing the host immune system, and the presence of syncytin in fetal stem cells helps them to escape destruction by the maternal immune system. A syncytin-like protein has evolved from endogenous retroviral sequences at least 3 times in other mammal lineages, such as mice, showing a clear evolutionary advantage.
Some viruses can even develop a more mutualistic relationship with a host or their hosts. The parasitic Echinummoid wasp has a close association with members of the Pollinator family. These viruses are capable only of the barest maintenance replication as proviral particles inside the wasp host. However, when a female wasp injects her eggs into a caterpillar, she passes the virus along with them. Now, in its preferred host, the virus kicks into high gear, suppressing development of the caterpillar and maintaining an ideal environment for the wasp's eggs. It suppresses development of the caterpillar, keeping it from developing into a butterfly until such a time as the wasp larvae hatch, killing their caterpillar wasp host. Female wasps leave with Pollinator proviruses incorporated into their germlines, and the cycle continues.
All right, so here is our paper, and in this 2016 publication, Groen et al. Wanted to investigate the effect of viral infection on pollinator attraction to plants. As the paper explains, Many plant viruses cause their hosts to express altered profiles of volatile organic compounds, or VOCs, that make the infected plant more attractive to vector insects such as aphids. However, the impact of virally induced changes in VOCs on pollinator species like bees is less well understood. In this paper, several experiments were done to determine the differences in the amount and type of VOCs produced by the infected plants and determine the bees' preference for infected plants versus healthy plants. The authors hypothesized that the difference in VOC production would attract pollinators to increase infected plants, increasing the plant's reproduction and partially compensating for the negative impact of infection on their fitness.
The unique VOCs produced only by the infected plants were isolated and identified by mass spectrometry. Free choice assays where the plant was covered up by a cup demonstrate that bumblebees show an innate preference for CMV-infected tomato plants over non-infected plants by sense of smell alone. The authors also included a test of bees' preference for plants infected with a mutant CMV whose gene for viral protein 2B was knocked out. Evidently, the 2B protein is crucial for suppressing antiviral RNA silencing. A mutant with a non-functional version of 2B was largely ignored by the bumblebees, and the normal CMV-infected tomatoes were instead pollinated preferentially. Tomato plants infected by CMV demonstrated stunted growth, although they did produce more flowers relative to their size than healthy plants. As shown in Figure 5, panel A, CMV-infected plants produce significantly fewer seeds than their non-infected counterparts. However, buzz pollination, as stimulated by the researchers with an electric toothbrush, did significantly improve the seed yield of the infected plants. While they didn't reach the production levels of non-infected plants, as can be seen in panel A, with pollination, they were able to recover to approximately half the levels of non-infected plants. Panel B is a comparison of the mass of the seeds, a metric of seed quality. Infection with CMV did not diminish the seed quality or the seed size of its host plant, and with buzz pollination, even achieved a significant yet small increase in seed quality over the uninfected. This suggests that infection does not impair the production of healthy seeds.
At the end of the paper, the authors used the data from the free choice assays and the seed yield experiments, as well as known data about tomato growth patterns and CMV infection, to generate a mathematical model. The researchers proposed a fitness trade-off between susceptibility or resistance to cucumber mosaic virus CMV. Infected plants did not grow as tall, potentially decreasing their ability to compete with resistant plants for resources such as sunlight, as well as a decrease in seed numbers, harming the plant's fitness as female parents. However, CMV appeared to increase the plant's attractiveness to pollinators through alteration of the VOC profile, as well as leaving seed quality untouched and increasing the number of flowers relative to body size. The author's model indicated that the increased attraction of bees to the infected plants could help offset the negative impact of infection on seed production by increasing the pollen yield. As a result, despite strong selection for CMV resistance, the genes for susceptibility remain common due to their reproductive advantage at attracting pollinators. The plants cannot evolve resistance without harming their reproductive output, and the virus maintains a susceptible host population large enough that it will be able to continue replicating in future generations.